AS level biology 9700. Students, we are going to start from first topic, which is cell structure. Before going in detail, I would like to discuss the three papers in which you have to appear. That is the P1, which consists of 40 MCQs, and marks for to solve these 40 MCQs will be 40, and time 1 hour 15 minutes. And the P2 will be consist of structured questions, and marks 60, and time will be the same 1 hour 15 minutes. And then P3 will be consist of 2 practicals, and marks for these 2 practicals will be 40, and time will be 2 hours to perform these 2 practicals. So it was all about this. Now we are moving towards the next slide. And in next slide, first of all, we will see what will be the part of the P3 in this first topic, cell. And that part is the microscope in cell studies. And this is the syllabus which is written in front of you on screen. So I will, you can say, discuss the syllabus of P3. Make temporary preparations of cellular materials suitable for viewing with a light microscope. Light microscope. It means you have to study about the microscopy, because there are 2 ways by which you can observe the cells. First of all, you will study the cell. You will study the different parts of the cells. Then when you are going to perform your practicals, and during the practicals, you must know what is microscopy, how to observe the cells with the help of light microscope, or how to perform the practical in the lab. So these are the, you can say, these are the different topics which you must know when you are going to study AS levels, because P3 is very important now, in which you have to perform the practicals. So here you can see the first, that is the MULA. With the help of light microscope, you have to prepare the slides, and you have to observe those slides under the light microscope. Then you have to draw cells from microscope slides and photo micrographs. That is, with the help of those slides which you will observe under the microscope, you have to draw them, the different cells or the different tissues which will be shown under the microscope. You have to draw them, the plain diagrams of those cells or tissues. Then calculate magnifications of images and actual sizes of the specimens from drawings, photomicrographs, and electron micrographs. That is, the different types of electron micrographs. It means you have to study the electron micrograph as well, because these are 2 major, you can say, by which you can observe the cells. Cells are unable to see with the naked eye. So we observe them with the help of light microscope, and with the help of electron microscope and light microscope. So these are the 2. And further, the electron microscopes which you have to study, they are scanning and transmission as well. So we will discuss that later in this chapter. Now, first of all, you have to calculate the magnifications. So magnifications, you will calculate the magnification with the help of these microscopes. Then next, use an eyepiece, reticule, and stage micrometer scale to make measurements, and use the appropriate units, millimeter, micrometer, and nanometers. Eyepiece, reticule, and stage micrometer. These are the 2 types of scales which you use during the magnification. And then next, define resolution and magnification and explain the differences between these terms with reference to light microscope and electron microscope. So you have to study resolution, magnification as well. So all these points which we have discussed right now, they can be the part of P3. They can be, the magnification can also be in P1 and P2, but mostly they can be the part of P3. So we have to study all these things, all these topics one by one. So first of all, we will start from the cell, and you know about what are the cells. So cells are the building blocks of all living things. All living things are made up of these cells. So the human bodies consist of trillions of cells. So they provide structure for the body, take in nutrients from food, convert these nutrients into energy, and carry out specialized functions. Cells have many parts, each with a different function. You know that these cells, when they join together, these cells, when they join together, or a group of cells to perform specific functions, is called as tissue. And many tissues, when they join together to perform the different functions, they will make up an organ. An organ will make up an organ system. An organ system will make up an organisms. So it means there are different types of cells which have to perform the different functions, because when they join together, when they make up a tissue, and the tissues will make up an organ, so they have to perform the different functions. They are specific for their functions. So they can you can say provide structure for the body. They can provide structure for the body, take in the nutrients. That is, when we take in nutrients, then those nutrients should be the part of the body after digestion and absorption. So digestion and absorption are also involved, and they have to convert these nutrients into energy for the process of respiration, or by the process of respiration, these nutrients, they become part of the body and they release energy. We have to get energy from these nutrients, and they carry out the specialized functions. What are the specialized functions which our body need to be performed? They can be the coordination and response. There can be the reproduction. So in this way, our body has to perform different functions, and according to the need of the body, our body needs specific cells to perform these specific functions. So this is the reason that human body is composed of trillions of cells. So these cells are performing these specific functions in the body. Now, when we study cells, that special branch in which we study cell is called as cell biology. So cellular biology or cytology is another name of this cell biology. So cell consists of the cells, all cells, whether they are animal cells or they plant cells. They have cytoplasm, and cytoplasm enclosed by a membrane. These are very all those informations which we have already discussed in O levels. So which contains many biomolecules as proteins and nucleic acids. All they are part of the cell. So most plant and animal cells are only visible under a microscope. They are very small in size. So they are very small in size, but if their dimensions are between 1 and 100 micrometers, they can be observed under the microscope. So already told you in the description, that is, there are two ways to observe the cells, because we need to observe the cells, how they are performing the different functions, what are their components, what are their parts, what are their you can say the requirements. So how they are performing the functions, we need to observe them. So there are two ways to observe the cells. So for this, we observe the cells by light microscope and electron microscope. So these are two microscope, types of microscope. First of all, we will go with the light microscope. So the light microscope is also called as the compound microscope, and this compound microscope or the light microscope um, is an instrument for visualizing fine details of an object. So this is to study the fine details of an object, which is kept under the microscope. So it does this by creating a magnified image through the use of a series of glass lenses. It means the magnified glasses or lenses, they are part of the light microscope. So which first focus a beam of light onto or through an object, and convex lens to enlarge the image formed. First of all, the image, there is need of a light, and that light will pass through the object, and then it will be focused, and then with the help of convex lens, it will be magnified, it will be enlarged, and then it will be visible in the eyepiece. So it means we need to know the different parts of the light microscope, which are on the next slide. And here you can see a small view of light microscope. So first of all, you can see it. We will start from the light source. Light source, which is here we can use a bulb or small bulb for to provide us the light. And then you can see the iris diaphragm, just like our eye, iris diaphragm, and then condenser. And then we will study the functions which are given on the right side. So glass slides, which is used to on the stage to observe the object. And then the cover slip should always be over there, and objective lens is over there. Objective is the have object lens, which is kept over the object which you have to study by the slide. So light beam, which will pass through this object and it will reach to your eye, and then with the help of eyepiece you can observe the specimen or the slide, which is kept for study. So here I would like to say the path will it can be a small bulb. So condenser iris diaphragm, condenser iris diaphragm is closed slightly to produce a narrow beam of light. So this is the function of this condenser iris diaphragm. That is, it will be, you can say, to produce a narrow. So it has very small hole through which the light is passed. Due to when the light passes through this condenser iris diaphragm, then it will produce a narrow beam of light. You can see a narrow beam of light. Then this narrow beam of light will pass through the condenser lens, which will focus the light onto the specimen. So the function of this condenser lens is to focus the light on the specimen, which is held between the cover slip and the slide. You can see the object will be there on the slide in the center of this glass slide, and cover slip will be over it to save it, to protect it. That is, the specimen should not fall away. Then you can say the light will pass through the specimen and then or this slide, or then through it will pass through the objective lens. So the objective lens will collect the light. It will, because the light can scatter in the other directions. So the objective lens will be used to collect the light, which will be passing through the specimen and produces a magnified image. You can see this light is moving in the upward direction. So then this beam of light will pass through the eyepiece or lens of the eyepiece, which will magnifies and focuses the image from the objective onto the eye. So it will be magnified, it will be focused, and then due to which we will be able to observe through this eyepiece. So in this way, you can see light microscope is working with the help of the lenses which are inside objective lens and eyepiece lens, which are there, which will be helpful to magnify all these images. So here we are going to discuss what is the advantage of light microscope. The one big advantage of light microscope is the ability to observe living things. That is, it means a very, very important point is there. That is, we can observe with the living cells. Living cells. Living cells means you can observe the cell division, the different phases of cell divisions. You can observe the different processes, as for example, osmosis and the cell division and other biological activities which you can observe, as for example, the uptake of the food by the cells. So in this way, you can see with the help of different techniques. Additionally, it is possible to use in vivo staining techniques to observe the uptake of colored pigments by the cells. So it means this is a very big, great advantage of the light microscope. That is, we are able the different activities of the living cells under the microscope. Otherwise, it is not possible. How can we observe? Cells are not possible to see with the naked eye. So only it can be observed in the light microscope. That is, we are able the different activities of the cell lively. So different biological activities, cell division, very important, as you know, which can be observed under microscope. So this is the main use of this light microscope. Now the next part which we have to discuss is the magnification. So first of all, magnification. Magnification is the to magnify the objects. When they will be magnified, then they will become larger than their actual size. When they will become larger from their actual size, it will be easy to study them. For this purpose, for the magnification in your syllabus, in O levels, when you were in O levels, you have studied the simple magnification. But now the magnification can be done with the help of this microscope, light microscope, in which you can use two special scales, which are called as eyepiece reticule, which is kept in the eyepiece. And the second is stage micrometer scale, which is kept on the stage where you need to keep the slide. So I would like to go first with the eyepiece reticule, which is a little scale bar that you can place in the eyepiece of your light microscope. Yes, in the eyepiece of your light microscope, you can keep, it can be made up of plastic or it can be made up of glass. So both of these materials can be used in the manufacture of this eyepiece reticule. When you look down the microscope, you can see the reticule as well as the specimen. It will be over the, as for example, if this is the cell and this scale will be over there. So scale will be over there. So with the help of the scales, you can measure them. The reticule is marked off in reticule units. So you can use the reticule to measure the specimen you are viewing in these reticule units. Just turn this eyepiece so that the reticule scale lies over the object you want to measure. You want to measure the whole object or the part of the object or the part of the specimen or the whole of the specimen, you can make use of this eyepiece reticule, which is kept in your eyepiece. And with the help of this, you can measure the actual length. So the other scale, which is a microscope stage micrometer, refers to a slide with the scale. I will show in the next slide. The slides are mounted with a reticle scale that is used for calibrating the eyepiece reticles as well as objective powers. So stage micrometer scale is always kept on the stage. Again, it is made up just like a scale. It is made up of glass, or it can made up of plastic as well. So I would I want to show. So you can see these two scales in this way. The upper one, which is eyepiece reticule scale, which has 0 to 100 units, whereas the stage micrometer scale, you can see it is about 0.012 or 0.01 equal to 1 unit. So I'm going with the next. So how can we make use of these two scales? How can we measure the cells under the microscope? So the scale over a human cheek epithelial cells, this say, I think I should show you this one. So here you can see this is eyepiece reticule scale. So cheek cells, if you want to observe these cheek cells, and these cheek cells are mounted on a glass slide, and that glass slide is kept on the stage. And here you can see you want to observe this cell. You want to observe this cell, you can see this cell. It is between 40 to 60, between 48 to 60. It means this cell has consists of 20 units. And if we calibrate the eyepiece reticule, and then this is the lower one is the stage micrometer. The scales, you can see it is equal to 0.01 or 0.1 millimeter. The unit of stage micrometer is millimeter, whereas the eyepiece reticule consists of 0 to 100 units, not scale. So in this way, I would like to say you that we have to calibrate these two scales to find out the length of this specimen set. How can we calibrate? As you can see, you will align these two scales. That is, eyepiece reticule, it's 100 units, and it is 0.25 millimeter. 0.25 millimeter, as it was just like this. You can see cell was just like this. It was 40 and on this 60. So, and it was 0.1 to 0.25. So it was about 0.1 to 0.25. You can see, I would like to go back in this way. You can see it will be equal to it. The cell was, if we cell and this way. So we are in this way. You can see now we are calibrating it. It means the 100 units are first of all, what is calibration? So calibration, you can say this is a process by which we align these two scales to get the accurate measurement, or to get the accurate or precise of the objects. So for this calibration, you can see here in this case, you can see 100 units are equal to 0.25 millimeter. So when we keep them, it is eyepiece, reticule, and this is stage micrometer. So it is both, they are in this way equal. So For calibrating them, 0.25, we will 0.25, we will divide it with 100 units. We will divide it with 100 units. So it will become 0. As for example, 0.25, if we divide it with 100, so it will be equal to 0.0025. This is the way of calculating or the calibrating them. So, or converting millimeter to micrometer, then we will convert this 0.0025 into micrometer. So for this, we have to multiply it with 1000. So when we will multiply it with 1000, so it will become equal to 2.5 micrometer per 100. So it will become equal to, so in this way. So the diameter of the cell shown superimposed on the scale in figure 20 eyepiece units, and so its equal diameter is, we will multiply that 20. It, we are talking about this, which was cheek cell between 40 and 60 units of eyepiece reticule. We will multiply those 20 units with the 2.5 micrometer. It will become 50 micrometer. So in this way, we have calibrated. So in all type of uh, magnifications in which you are making use of eyepiece reticule or stage micrometer, you have to perform this function for calibration. So you will calibrate, then you will find out the magnification. In the next, I have taken this example from your book, that is calculating the magnification of a photograph or image. This is very simple magnification which already studied in O levels. That is, in step 1, the real length of the cell is 80 micrometer, and step 1, measure the length in millimeter of the cell in the photograph using a ruler. You should find that it is about 60 millimeter. So this is about 60 millimeter if you have measured the photograph, and then 1 millimeter is equal to 1000 micrometer. And because you need to convert the scale, you are not to give the answer in millimeters. You need to convert the millimeter into micrometer. So we will convert the millimeter into micrometer. So it is about 60 millimeters. So we will multiply 60 with 1000. So it will become 60,000 micrometer. Because micrometer is a smaller scale than the millimeter. So we will multiply it with 1000. So use the equation. We will use this equation, simple equation of magnification, which is equal to image size divided by actual size. So we will, so this is the actual length and this is the image length which we have measured from the photograph which they had given us. So it will, when we divide, so it will become, the magnification will become 750. Well, students, it was all about today's class. Please like, share, and subscribe my channel. Thanks for watching.